Hello friends, I Dr. Minal Harde from Department of Pharmaceutical Chemistry, PE Society's Modern College of Pharmacy, Nigri, Pune. Welcome you all for today's session. Today we are going to see details on topic Atomic Absorption Spectroscopy. Atomic Absorption Spectroscopy is a technique in which we are determining metals as well as non-metals. So today we are going to see content such as introduction, theory, instrumentation and application. To start with introduction, what is atomic absorption spectroscopy? Atomic absorption spectroscopy is a quantitative method of analysis that is applicable to many metals as well as to non-metals. It was introduced by scientist Alan Walsh in 1950s. This is the most powerful technique for quantitative determination of trace metals in liquids. This method provides total metal content of sample and is almost independent of molecular form of metal in the liquid. Example: cadmium content of water sample. It does not matter whether the cadmium exists as a chloride, nitrate, sulfate or other salt. Versatility of atomic absorption spectroscopy. It is helpful to determine 60 to 70 elements, including most common rare earth metals. It determines from few concentration ranges till micro quantities. It is helpful for determining the non-metals also by indirect method. Atomic absorption spectroscopy is restricted to aqueous as well as non-aqueous solutions. Atomic absorption spectroscopy does not demand sample preparation. It is the ideal tool for non-chemists also. Example: engineers, biologists or clinicians are interested in the significance of results. That's why it is the direct tool also for this another field. It is a very sensitive technique. It detects very low quantity as well as it is lower than 1 ppm it detects. Disadvantage of this technique is only one element can be determined at a time. A change in source and change in wavelength is necessary to determine second element. Principle of atomic absorption spectroscopy. Atomic absorption spectroscopy is based on absorption of energy by an element. Element absorb the energy radiations at a ground state in the gaseous form of atomic absorption spectroscopy. When a solution containing metallic species is introduced into the flame, the vapor of metallic species will be obtained. Some of the metal atoms may be raised to an element level, energy level, sufficiently high to emit the characteristic radiation of the metal. But the large percentage of metal atom will remain in non-emitting ground state. This ground state atoms of particular element are receptive of light radiations of their own specific resonance wavelength. Thus, when the light of this wavelength is allowed to pass through the flame having atoms of metallic species, part of the light will be absorbed and the absorption will be proportional to the density of atoms in the flame. Thus, in atomic absorption spectroscopy, one determines the amount of light absorbed. Once this value of absorption is known, the concentration of metallic element can be known because absorption is proportional to the density of atom in the flame. Atomic absorption spectroscopy is helpful for detection of non-metals also. The energy required for excitation of non-metals are very high, so that non-metals are detected by indirect method. Example: chlorides can be precipitated as silver salts. From the subsequent determination of silver, the chloride can be detected. To start with instrumentation, this is the schematic diagram of the atomic absorption spectrophotometer. Here first is the line source, this is the radiation source. Then flame, after that monochromator, detector and readout system, where the nebulizer is attached to the flame. To start with the instrumentation part, this is the diagram. Here SCL is written, which is the source of the radiation. This source of radiation is passing through the burner or flame, and then it is bombarding on the monochromators, and it is the concentration of metal is detected by the detector. This is the proper or actual atomic absorption spectrophotometer. This is the diagram of instrument of atomic absorption spectrophotometer. Then we are starting with the radiation source. What should be the ideal properties of the radiation source? It should be stable, it should be intense, as well as whatever the resonance line of element we are getting, it should be stable. Preferably the resonance spectral line should be narrow as compared with the width of absorption lines to be measured. So the source include hollow cathode lamp. What is hollow cathode lamp? Hollow cathode lamp, here it contains anode, hollow cathode, glass shield, neon or argon gas, as well as quartz or pyrex window. This is the actual picture of the hollow cathode lamp. Now, construction of hollow cathode lamp. The cathode lamp consists of hollow cup. In the cup, element is present which has to determine. For example, to determine sodium, sodium lamp is required. Another is the tungsten wire, which is the anode. The two electrodes are housed in a tube containing an inert gas. The lamp window is constructed of either quartz, silica or glass. The exact material depends upon the wavelength which has to be transmitted. Operation of hollow cathode lamp. So how this hollow cathode lamp works? When the direct current voltage of 300 to 500 volt is put across the anode and the cathode, the atoms of the filler gas undergo ionization at the anode and are rapidly attracted by the cathode. Then the fast-moving ions strike the surface of the cathode and physically determine and displace the surface metal atoms of the cathode into the argon or neon gas. Further, collision of vaporized metal atom from the cathode with energetically filled gas ions resulted in excited metal atoms which emit the characteristic spectrum of the metal used to construct the cathode. Neon or argon gas filled in hollow cathode lamp performs three functions. It is the main source of current carrying capacity in the hollow cathode lamp. It dislodges atoms from the surface of cathode. It is primarily responsible for excitation of the ground state metal atoms. The pressure maintained in hollow cathode lamp is between 1 to 5 torr. If higher pressure is maintained, the discharge tends to be unstable, and if lower pressure is maintained, then vaporization of hollow cathode metal increases and operation temperature increases. That's why we have to maintain 1 to 5 torr pressure. The spectral lines produced by hollow cathode lamp is so narrow that they are completely absorbed by the atom. By this method, one can easily detect and measure the atomic absorption. Each hollow cathode lamp emits the spectrum of metal which is used in the cathode. For example, copper cathode emits copper spectrum which is only absorbed by copper atom. Zinc cathode emits the zinc spectrum which is only absorbed by zinc atom, and so on. In atomic absorption spectroscopy, gaseous discharge lamp, it is also called as arc lamp, is also used. This lamp contains an inert gas. At low temperature, and the metal or metal salt. These lamps are useful for the alkali metals zinc, cadmium and mercury. Next is chopper. What is chopper? So after line source or radiation source and between the flame, chopper is present. The rotating wheel is interposed between the hollow cathode lamp and the flame. This rotating wheel is known as chopper and interposed to break the steady light from the lamp into an intermittent or pulsating light. This gives a pulsating current to the photocell or detector. There is only a steady current caused by light which is emitted by the flame. But only the pulsating or alternating current is amplified and recorded, and thus the absorption of light will be measured without interference from the light emitted by the flame itself. Next is burner. Sample solution, the fuel and oxidizing gases are passed through separate passages to meet at the base of flame. Flame breaks up the liquid sample into droplets which are then evaporated or burned, leaving the residue which is reduced to atoms. Total consumption burner do use oxygen with hydrogen or acetylene and give very hot flame. Extra turbulence in the flame from variation in droplet size increases noise. Next is the premix or laminar flow burner. This burner, in this, sample, fuel and oxidant are mixed prior to entering the flame. Turbulence drastically reduced by removing larger droplets. Mixing baffle ensure only fine mist. Premix burner is very suitable for atomic absorption studies of metals of group 1A, 1B and 2B. Next is monochromator. In atomic absorption spectroscopy, most monochromators are prism or grating. The function of monochromator is to select a given absorbing line from spectral lines emitted from the hollow cathode lamp. For many elements, higher dispersion is necessary. When the cathode lamp is hollow cathode lamp is made up of transition metal, the emission spectrum from hollow cathode is so complicated that high dispersion is essential. For such cases, large dispersion and high resolution are advantageous. Then detector. Mostly the photomultiplier detector is used in atomic absorption spectroscopy. This is the very well-known detector. Here in this, diodes, that is the series of cathodes as well as anodes are attached, where the radiations are bombarded on the cathode and then multiplication of the removed electrons is happen on the diodes, and number of electrons is absorbed by the anode, and whatever the current is amplified, on the basis of that quantitative analysis can be performed. After that, readout system. Readout system is generally the potentiometer or pen recorder. Nowadays we have the computer screen to record the result. Application. This technique is used for qualitative analysis, quantitative analysis, simultaneous multi-component analysis, as well as for determination of metals in biological materials, determination of metallic elements in food industry, determination of calcium, magnesium, sodium, potassium in blood serum, as well as for determination of lead in petrol. Thank you very much.